Thank you.
So shall I start?
Sir, I will start with introduction, then I will give it to you, sir. Um, just waiting for the clock to hit 10 so that people who are on time will be respected. Very few people are like that. So, hello everyone. Thank you for joining us today. And those who will be seeing this particular recording at a later time, hello to you also. Today, we have one of the prominent personalities in the emerging biotherapeutics field, Dr. Ajay Singh, who is the Assistant General Manager at Genova Biopharmaceuticals. Dr. Ajay Singh is a PhD in biochemistry from Delhi University South Campus, and he has been associated with Genova Pharmaceuticals since 2013. He has risen up the rank. He has started from an executive position to now an assistant general manager position in Genova Biopharmaceuticals. So I presume that he has seen all the different challenges at various levels and also the challenges in developing newer type of therapeutics very much relevant to India. Um, the Genova Biopharmaceutical is a company which is headquartered in Pune. It's a biotechnology company dedicated in developing, producing, and commercializing the biotherapeutics to address the life-threatening diseases across various indications. And recently, they were in the news for developing the Jumbo Vac 19, that is Jumco Vac 19, which was a mRNA vaccine for COVID-19. And Dr. Ajay Singh has been at the forefront of developing this particular molecule, all in India, fully indigenously designed and developed in India. So I look forward to hearing from Dr. Ajay Singh. Over to you, sir.
Uh, thank you, Dr. Rao, for this exciting introduction. And good morning, everyone. And especially special thanks to ORD for inviting me for this talk. So let me share the screen. Yeah, so I guess everybody can see my screen. Uh, so as Dr. Rao said that I am in Genova from 2013. So I did my PhD almost 12 years back, and since then I am associated with Genova. And the core area of my research is the mRNA vaccine, cell and gene therapy, and also the antibody engineering. mRNA vaccines or mRNA is a not new concept. The basic research regarding the mRNA was initiated almost 50 to 80 years back, but now the fruits are coming out from this research. And recently, this mRNA vaccine has gained a lot of attention during the COVID, and also there are a lot of clinical trials and also publications, full publications which are reporting mRNA as a very promising therapeutic agent. So today I'll be talking primarily on the mRNA future or achievements in the field of vaccinology. So here is the overview of my presentation. So first I'll be talking a bit about the company overview, that is Genova Biopharmaceuticals Limited, Pune. Just a minute. Yeah. So Genova Biopharmaceuticals. So we are based of Pune, Maharashtra. And then uh, I have a couple of slides for the Genova recent success, that is using the cutting edge technology, which is mRNA. As Dr. Rao said, we are the first one in India to launch the mRNA vaccine. And next, I'll be talking about the mRNA vaccine portfolio in detail. What exactly portfolio mean to us, what all things we are covering in the portfolio. So everything I'll be talking in details. And finally, a couple of focus area for Genova for next 3 to 4 years. And then finally, it will be the take-home message from my end. Coming to the company overview, so Genova Biopharmaceuticals was established almost 15 years back, and we are based off Pune, Maharashtra. And so for the ease of understanding, I have put 3 questions here. So what we do, how we do, and why we do. So what we do is we manufacture biotherapeutics and mRNA vaccine. And next question is how we do. So we do this by developing newer platform and innovative biomanufacturing technologies. And finally, why we do so the objective or the prime focus for us is to transform the healthcare using biotherapeutics which are developed or manufactured by using innovative platform or technologies. We are based off Pune. Our headquarters is in Pune. So coming to the team size, so Genova has close to 650 employees. Out of them, 116 are at R&D, and rest of them are at manufacturing. We have almost 24 PhDs which are working on in different parts or different verticals at the Genova, covering from research to manufacturing and and to the marketing as well. And we have candidates who have got trained from various prestigious institutions across the globe like NIH and almost all possible institutions. And coming to the gene to market, so we have our own facility or own capabilities of developing the clone, followed by the cell banking, expression studies, process development, analytical characterization, and the fill finish. So in short, you can say that we have entire capacity to bring the product from gene to the market. We have our own intellectual property team, IP team, and at R&D we have couple of unique expertise like biomanufacturing practices using the AI-based approach, and for mRNA, cell and gene therapy, and antibody engineering, most of things we have mm, introduced the AI-based approach in all these things. And manufacturing, we have couple of manufacturing units, again based in the Pune, Maharashtra only. So we have the capacity to make the bulk drug products and mRNA vaccine. For fill finish also, we have the capability for vial and PFS both, and liquid and lyophilized in both the presentation. We have our own quality team where we have our own system to make the method development, to, to develop the unique method and validate the method. We have our own team for clinical trial, regulatory and marketing. For the vaccine program, we have many collaborations across the globe, and these collaborations are primarily focusing on the malaria, tuberculosis, leishmania, COVID-19 vaccine, flu vaccine, and also we have developed a lot of adjuvants which are used for the various vaccines globally. Coming to the focus areas, so this is a standard diagram of the central dogma. So we start with the DNA molecule, and from DNA, it goes into the mRNA by the process of transcription. And this mRNA is a very transient molecule that finally make the protein, which is by the process of translation. So every information in the body is stored in the form of DNA. And whenever the action is required, the mRNA is made in a transient nature. And finally, this mRNA is translated into the protein, that is the action component. So Genova has, coming to the protein side, so Genova has two platforms, mammalian and microbial platform. From mammalian platform, we have three molecules which are commercialized since last more than 10 years. We have tenecteplase that is given for AMI, and also we have got this product approved for the AIS. Similarly, we have the erythropoietin, which is given in case of CKD related anemia. So both these products or these three products are approved since more than 10 years. And for many of them, we are the market leader, and tenecteplase is a kind of life-saving drug. In the pipeline, we have bevacizumab for the wet AMD. So these molecules are coming from the mammalian vertical, and from microbial vertical, we have three products commercialized. That is GCSF for oncology, PEG GCSF again for the oncology, and PEG asparagase for the ALL. These are our commercialized products, and two new products which are in development or in the advanced stage of development. Sooner we are entering into the clinical trial for these. So that is the asparaginase, and the ervinase. With the success of protein side, we entered into the mRNA side, and at mRNA level, we have recently launched our mRNA vaccine, or first mRNA vaccine, that is Gemcovac 19, which is for COVID-19. And multivariant vaccine for COVID is under development. We are also developing various vaccines like vaccine against the monkeypox, zoster, HPV, dengue, and malaria. And many of them are at the late stage of development. So maybe in next couple of years, we will be having these vaccines in the market. And with all these success from the mRNA side and protein side, we have entered into the cell and gene therapy area, and we are hoping that in maybe by 2023 and or maybe by early 2024, we should be having some good news to share. Now coming to the second category of my talk, that is the Genova most recent success using the cutting edge technology. So the vaccine that we have made is the GEMCO-Vac 19, which is mRNA vaccine for COVID-19, and the EUA, that is emergency use authorization, was granted in India on June 28, 2022. This vaccine has been listed in the COVID app as well. Sorry. So mRNA vaccine for COVID-19, it's first indigenously developed mRNA vaccine in India that has received the EUA from the Office of DCGI. It's also the first EUA granted thermostable lyophilized mRNA vaccine in the globe, because before this vaccine, there are two mRNA vaccine, one from Moderna and another from Pfizer are in the market, but they are having the sub-zero storage temperature. That is the biggest challenge for mass deployment of those vaccines. The vaccine that we have made is entirely thermostable, that can be stored at 2 to 8 degrees centigrade, and we have the stability data of more than a year right now. And next is its first EUA granted self-replicating mRNA vaccine. So mRNA vaccines are of two kinds. One is the self-replicating and another one is the non-replicating. So the Moderna and Pfizer vaccines are the non-replicating mRNA, though the Genova that we have made is the self-replicating, which has various advantages, especially in terms of dose regimens. So the vaccine from Pfizer is given at the dose of 30 microgram and Moderna is given at the dose of 100 microgram. But the vaccine that Genova has made is given at the dose of 10 microgram. And finally, it's the third EUA granted mRNA vaccine for COVID-19 in the globe. As I said, first vaccine was the Moderna, followed by the vaccine mRNA vaccine from the Pfizer, and third one is from the Genova. So GEMCO-Vac 19 has two components. One is the mRNA encoding for the spike protein of SARS-CoV-2 virus, and another is the cationic lipid nanoemulsion, which is used as a carrier molecule to deliver the mRNA inside the cell. And the funding support was provided for this development was from DBT. So we received approximately 25 crore as a seed funding from ENDOCEPI mission and 100 crore for the scale up and CT from the COVID Suraksha Fund. Here are the clinical outcomes. So we did the phase 1 study in 87 volunteers, phase 2 in 415 volunteers, and that has established the safety. Phase 3 study was conducted in 4,000 volunteers. I'll go into the details in next couple of slides. So here we have used the Covishield as our comparator, and the immunogenicity of GEMCO-Vac 19 was found to be comparable with the already approved Covishield vaccine. Now coming to the most differentiating factor of the GEMCO-Vac 19. So GEMCO-Vac 19, as I said, it's a thermostable, which is a first thermostable mRNA vaccine that has received the EUA. So if you see how we have developed the thermostability, so the basic difference between DNA and RNA is the sugar moiety. So in case of DNA, the sugar is deoxyribose sugar, which has the hydrogen bond, but in case of RNA, the sugar is having the hydroxyl bond, and this hydroxyl bond is the main agent of degradation of mRNA, and that's the reason why mRNA vaccines are stored at sub-zero temperature, that is nearly -20 to -40 to -80 degrees centigrade. Understanding this concept, we remove the water content entirely from the vaccine, and that's how we have made our vaccine thermostable at 2 to 8 degrees centigrade. This OH group play a very important role in maintaining the phosphodiester bond, which is the backbone of any nucleic acid based chain, that is either could be DNA or mRNA. But this OH group is hydrolyzed in the presence of water, and that's the main reason for the degradation of mRNA. Now, so this is how our vaccine looks. It's lyophilized. So here is our cake. And if you see the immunogenicity data, so this graph is representing the anti-spike protein IgG titer. Blue one is the vaccine, which is the liquid that is stored at -80 degrees centigrade, and green one is the vaccine which is lyophilized. So if you see the day 14, day 28, and day 43 data, this data is from animal, and compare this data with the lyophilized version again from at day 14, day 28, and day 43. So there are no significant difference in the IgG titer in the vaccine which is coming from the frozen condition or the vaccine which is coming from the lyophilized condition. And if you see the surrogate neutralization potential, so again there are not much difference, and moreover the lyophilized is showing quite higher neutralization compared with the frozen version at day 28. And this data set suggested us to move ahead with the lyophilized version. So the vaccine that we have got the approval is the lyophilized. So coming to the preclinical study summary of the Gemcovac 19. So Gemcovac 19 immunogenicity was checked in mice, rat, and hamster. Toxicity we checked in the rabbits. And for challenge study, we used the hamster model. And this study was conducted at ISC Bangalore. And our vaccine was found to be safe, and also the vaccine was found to reduce the clinical sign and lung tissue pathology in case of challenge study. Here is our phase 1 concert diagram. So we screened 364 subjects. This study was done at the time when India was struggling with the second wave of COVID infection. So the failure rate was very high because we were looking for the seronegative individuals. So we screened almost 364 individuals, and we got only 82 individuals which were seronegative. These 82 were then segregated into two groups, that is 18 to 55 and 56 to 70. And each group received 3 doses, 5 microgram and 10 microgram and 25 microgram of the mRNA. So phase 1 summary, the safety was established in the safety was established, and for Gemcovac 19 was well tolerated at 5 microgram, 10 microgram and 25 microgram dose, and there was no adverse event observed in case of Gemcovac 19. Coming to the immunogenicity side, so found this Gemcovac 19 was found to be immunogenic both at the humoral and cellular level, and 10 microgram dose has given better immune response compared with the 5 microgram, but it was quite equivalent to the 25 microgram, and that's how we chosen 10 microgram for our phase 1 phase 3 study. So phase 2 phase 3 study was a combined prospective multi-center randomized active control observer blind study, which was seamlessly followed by the phase 3 study to evaluate the safety, tolerability, and immunogenicity of the Gemcovac 19 candidate in healthy subjects. And here is our design. So for phase 2, we took the 400 subjects, and 200 were given in case of Gemcovac 19, and rest 200 were given in case of Covishield, and each category 30 subjects were taken as a seronegative, and then second dose was given at day 29. And after the 36-day safety, we immediately went for the phase 3 study, where we took the 4,000 subjects. Out of 3,000 subjects were in the Gemcovac 19 category, and rest of the 1,000 were in the Covishield category. And based on the data set, we got the approval for our Gemcovac 19. So here is the summary of phase 2. So Gemcovac 19 was generally safe and well tolerated. Solicited events were greater in case of Gemcovac 19, but it was restricted to grade 1 and grade 2. No difference in unsolicited events were found in comparing both the vaccine. No related serious adverse event were noted in case of Gemcovac 19. Death was found in one participant, which was who have received the Covishield due to pulmonary embolism. Coming to the immunogenicity section, so anti-spike IgG antibody titer were comparable in both the groups, that is non-inferior, our Gemcovac 19, and neutralizing antibodies were also comparable among both the groups. Both vaccines showed the Th1 bias response and no Th2 effect. These were the sites that we chosen for the phase 3. So we selected 36 sites, and the highlighted sites are the DBT ICMR clinical network site, and you can see that our sites were distributed across the India. Summary of the phase 3. So here we took 3,200 subjects who received the Gemcovac 19 Safety data set was established in case of 3,000 subjects in accordance with the FDA and WHO guideline. Gemcovac 19 was found safe and well tolerated. Up to 4 months, we have the data, and rest of the data are coming now. And solicited events observed in Gemcovac 19 were generally of mild and moderate category. No adverse event was observed in case of subjects. Gemcovac 19 generated robust immune response. Anti-spike IgG antibodies were non-inferior in Gemcovac 19 compared with the Covishield. Seroconversion rate was non-inferior in Gemcovac 19, again compared with the Covishield. Neutralizing antibodies, that is, we checked the neutralization using the PRID50, CPAS, and pseudovirus assay, were again comparable in both the groups. And Th1 bias in cellular immune response was observed in both the groups. Now, coming to the most exciting area, that is the booster. So almost more than 90% population have been vaccinated. So because the Gemcovac 19 was approved for the naive individuals, so we immediately went for the booster as well. So here we have made the Omicron specific booster, because right now Omicron is in the Omicron variant of the SARS-CoV-2 is into the circulation. So the route of administration here we have changed to intradermal. Earlier for Gemcovac 19 it was intramuscular, and this Omicron specific mRNA vaccine could be used as a heterologous booster over other vaccines. And regulatory submissions for this vaccine we are hoping by January 23. And this was our phase 2 and phase 3 design. For phase 2 we selected 140 subjects, and phase 3 we have taken 3140 subjects. Day 29 data is coming, and because the entire we have I guess by early this month we have finished our entire injection. So the day 29 data is coming, and as I said, by January 2023 we should be having the data for the regulatory submission. Now coming to the next category, that is mRNA vaccine portfolio. So for the ease of understanding, I have divided the vaccine platform which are available into the 4 categories. First is the vaccine 1.0, that consists of whole organism either in the live or weakened form. And vaccines are not new concept. Vaccine was invented almost 200 years back, but since in last 200 years we have seen numerous various upgradation in the platform. So vaccine 1.0 you can say is the most traditional platform, and vaccine 2.0 with the challenges of vaccine 1.0, vaccine 2.0 was developed, which is majorly focused on the subunit vaccine, consist of either protein, antigen, or protein components. And the best examples are seasonal influenza, hepatitis B, or HPV vaccine. Vaccine 3.0 is based on the adenoviral or plasmid DNA platform, and the best example are VSV Ebola vaccine or COVID-19 vaccine. And finally, the vaccine 4.0, that is entirely synthetic in nature, which is the mRNA vaccine, which does not require any kind of host for its propagation. As I said, it's entirely synthetic in nature. And if you see the process economics and time to manufacture, the time to manufacture is highest in case of 1.0, because for 1.0 you are dealing with the live organism, and sometimes people who are working on the ground are always at risk for getting contaminated or infected with the disease. And to manufacture these vaccines, either BSL-3 or BSL-4 kind of facilities are required. But as I said, vaccine 4.0 is completely synthetic in nature and does not require any host for its propagation. So no such standard GMP facility is only the requirement for manufacturing 4.0 kind of vaccine. And if you see the process economics also, so it is quite easy and economic to manufacture the vaccine 4.0, because no special kind of requirements are there. Establishing, now coming to the mRNA platform technology. So if you see this diagram, so it shows the potential of mRNA to enter into the clinical trial within the day 50 of the reporting of the first index case. And this timeline has already been proven during the COVID-19 for mRNA vaccine from Moderna, and also we have also developed our own Omicron specific vaccine within I guess 35 to 40 days. And if you see the so Genoa has developed and established the mRNA platform technology, and entire end-to-end development regarding this platform we have done in-house. And also we have done the backward integration that has ensured the supply of key raw materials. So backward integration means mRNA manufacturing process uses a lot of key raw material, especially the enzymes. We have our, since as I said in the beginning, we have our expertise in the protein side. So we have made all the enzymes in-house, and we are very minimally dependent on the vendors or the suppliers from different parts of the globe for these enzymes. Now coming to the portfolio in detail. So portfolio or mRNA portfolio has four components for us: mRNA vaccine, next is the recombinant protein, followed by the pseudovirus and surrogate-based neutralization. So for mRNA vaccine, so the whatever if we are given a disease to make the mRNA vaccine, so the gene is designed and cloned into a plasmid, and from this plasmid we make the mRNA by using the in vitro transcription method, and this mRNA is finally complex with a carrier molecule and finally this is ready to be injected. Next is the recombinant protein side. So for any vaccine, if we are making, we also need the recombinant protein so that the vaccine, which are or the mRNA vaccine after the human or animal injection, the sera should be tested to check the neutralizing antibodies and antibody titer. Now the pseudovirus. So we have developed our own HIV-based pseudovirus system to see the neutralization. And also the surrogate virus also we have developed our own in-house. So as I said, from the mRNA vaccine, if the sera is coming, so this sera can be tested on the protein which is coming from the recombinant protein section to as a bait to test the antibody titer. And also these pseudovirus and surrogate virus can be used to estimate the neutralization. So vaccine in detail. So here is our process flow. So here I have used the SARS-CoV-2 as a model to represent our vaccine portfolio. So this is the entire genome of the SARS-CoV-2, which is approximately 29 kb. From this molecule, we are using the spike protein as our target antigen. So we clone this spike protein into a plasmid, which is having various essential components for making the mRNA. As I said in the beginning, we are dealing with the self-amplifying. So along with this antigenic acid, we also give certain non-structural proteins which help in the self-replication of the mRNA construct. From the process of in vitro transcription, mRNA is made, and then this mRNA is complex with the nanoparticle that is a carrier molecule, and finally it is called as a mRNA vaccine. So these are the steps which are involved in for making this vaccine. First is the designing, followed by the cloning, then lead identification, because here we design a lot of constructs. We do a lot of codon optimization and harmonization to get the better immune response. And we have a method that I'll be discussing in detail in next few slides. So we do the lead identification, and the best lead is followed by the cell banking, and finally the mRNA drug substance and drug product manufacturing. Here is the process flow for the mammalian expression plasmid. So again, it starts with the designing, cloning, lead identification, followed by the banking and transient expression. Because if we are going for the CHO or XP293, we generally prefer to go with the transient expression. But in case of bacteria, we go for the stable expression, followed by the protein purification and characterization. Pseudovirus neutralization. So as I said that we have developed our own pseudovirus system based on the HIV lentiviral platform. So these are the plasmids which are used for making the pseudovirus, and this is the main plasmid where the desired gene is inserted, that is finally translated and present on the or these viruses are then pseudotyped with these proteins. So in case of COVID-19 or SARS-CoV-2, we are using the spike protein. So all these pseudoviruses are having the spike protein on their surface. So after the pseudovirus manufacturing, if we mix the pseudovirus with the sera which is having the neutralizing antibodies, so we get difference in the luciferase activities, because here luciferase is used as a reporter system, and based on the deferred activity of the luciferase, we have a mathematical model to calculate the neutralization potential. Now is the surrogate virus neutralization. So pseudovirus has certain challenges because you need a defined control facility to make the pseudovirus, but this surrogate virus can be made, and this assay can be performed in a standard lab practice. The only innovation that we have done here, the commercially available pseudovirus system are based on the chemical conjugation, but here we have made a recombinant fusion of these HRP, which is the reporter gene, with our probe substrate. So this mRNA manufacturing is a disease agonist platform. So mRNA, or for mRNA manufacturing, we need couple of reagents or raw materials like CLNE, which is the our carrier molecule, and then we need the enzymes and various cofactors. Rest, if you see the process, only plasmid get changed and rest of the things remain same. So if we have a plasmid specific for the SARS-CoV-2, at the end we will get the COVID-19 or the vaccine which is against the COVID-19. Now, if we just change the plasmid with some other disease, we will get the vaccine at the end. So here I am showing the example of zoster vaccine. That this vaccine also we are developing. Now, if you just change the plasmid, because rest everything remains same, even the process also remains same, you get the vaccine for the malaria. And if you change, you get the vaccine for the HPV. Similarly, if you change, you get the vaccine for the biotherapeutic. So biotherapeutic is again a very promising area where we are focusing, and this disease agonist feature is a very important feature for this mRNA because it reduces the development timeline for any vaccine or therapeutic which is coming out of this mRNA platform. Now, for the ease of understanding, I have divided this entire mRNA manufacturing into the 4 units. So first unit is the plasmid DNA. So here because plasmid only introduce the specificity for the vaccines. So plasmid DNA is manufactured and stored at the -20 temperature, followed by whenever the mRNA has to be made. So this plasmid DNA is used for making the mRNA drug substance, where this drug substance is stored at -80-degree temperature. Next is the CLNE, that is the carrier molecule. So this carrier molecule is also made independently and stored at 2 to 8 degree temperature. And whenever we need the vaccine, this drug substance and CLNE are mixed together in a defined ratio under defined condition to make the mRNA vaccine, which is a lyophilized. So these are the 4 units of unit operation to make the mRNA. We have done various innovation, and all of these units are scalable. So that's what we have done. And as I said, that we have done the backward integration. So here you can see for plasmid DNA, we have our own in-house production unit which make the plasmid. For the drug substance also, various enzymes are required. So we have our own facility to make the key enzymes and cofactor in-house. For CLNE also, there are various lipid molecules which are required. So we have our own facility to make the lipid as well. That has reduced the dependency. And finally, the vaccine, using all in-house material, we make the vaccine that is made in India for India. Now the challenges in democratization of the platform. So if you see, for bringing this platform to the low and middle income countries, there were various challenges for this mRNA platform. First is the ultra low temperature logistic, followed by the ease of manufacturing, then scalability, then supply of raw material, and finally the tech transfer to the regional hubs. So here, what differences we have brought here? So for ultra low logistic, we have made our vaccine stable at 2 to 8 degrees centigrade. For the ease of manufacturing, so we use a chemistry that for the complexing of mRNA to the carrier molecule that is readily scalable, and that's how we have reduced the dependency and losses and this has brought the ease for our manufacturing. Next is the self-replicating mRNA platform. So self-replicating, in case of self-replicating, we get the advantage of dose regimen. So this is again one advantage. And next is the backward integration. So using backward integration by making our own in-house enzymes and key raw materials, we have reduced the dependency, and now we are ready for the regional hub tech transfer. So here is the mRNA-based therapeutic market estimate. So if you see this graph, A graph, so it is showing the details of the industries in the form of industry or academia which are involved in the mRNA manufacturing. So in case of, so it has the data for the North America, Asia, and Europe. So if you see the private industries are on top for this mRNA. B is showing the platform wise segregation. So self-replicating and non-replicating platforms are of the major choices. And if you see the carrier molecule, so majorly all the mRNA vaccines are using the lipid nanoparticle, but also there are few molecules, which are few approaches which are using the naked mRNA. If you see the disease-wise segregation for this mRNA, so SARS-CoV-2 is the major focus area, followed by the flu and Zika. And these are the number of manufacturing sites which are present across the globe. Highest is in case of Asia, followed by the North America. As on today, the mRNA therapeutics are estimated more than $5,000 million market size, and it is estimated to reach more than 25,000 by end of 2030. So mRNA, so these RNA-based therapeutics are expected to remain dominant, considering both chronic and acute diseases. Coming to the focus area. So focus area 1. So we have developed our mRNA in-house, and this picture is showing the NAR, means the non-amplifying mRNA platform, which is having the 5' cap, followed by the 5' UTR, then ORF, and 3' UTR, followed by the poly A tail. And next, we also have the self-amplifying, where these NSPs are present other than these modules, which are non-structural protein and helps in the self-replication of the mRNA. Sorry. So if you see the application, so far we have touched the base of vaccine, and we have also entered into the booster side. Now we are entering into the therapeutic, transient protein expression, biotherapeutic, and cell and gene therapy. So this platform has to be reprogrammed, and that's what we are doing here. We are doing right now. So for cap incorporation, we have two methods. One is enzymatic, another is clean cap. For UTR, we are developing a UTR library, because it is not always wise to use a single set of UTR for all sort of vaccine, booster, or therapeutics. And for NSP also, to further increase the longevity of the expression, we are mutating. You can say that we have identified few hotspots in the non-structured protein, and those changes we are bringing here to make our mRNA long-lasting. And for increasing the expression side, we have done the codon harmonization. 3' UTR also we are we have made the library, and for best expression we are panning or selecting the best suitable UTR for our gene of interest. And again, we have various poly A lengths also. So here is the pipeline for the lead mRNA candidate identification for us. So we have developed a pipeline where we get the mRNA lead identification in just 30 days. And these are the certain stages that need to be performed. So this pipeline starts with the source and design, where we use a lot of portals or the database for identifying the gene of interest, followed by the gene design and assembly and cloning, plasmid, and mRNA. So we design a lot of construct, and then we have a in vitro cell-based assay to see which construct is giving better response, and based on that we identify our lead candidate. Now we have also developed the machine learning for designing the next generation mRNA. So this table is showing the 5' UTR and 3' UTR length from different species. So that's why, and now if you see the here in case of human, they have taken into account of 1200 sequences, and the maximum length of the UTR is 2800, and the minimum is 18 base pair. So that's the reason it is not always wise to just stick with a single UTR. And understanding this concept, we have developed the UTR library, and we always go for a best UTR selection. And here is our process. So UTR means the regulatory element library. We make the mRNA, we do the in vitro expression, we go for the polysome profiling, and mean ribosome load is identified based on our predictive models. And this gives the final understanding that which UTR is best among the our UTR library. Now the second focus area. So here our main focus is the CAR-T. So these are the list of the approved CAR-T therapies. And if you see the cost, so the cost per patient goes from the 450 to 750,000 USD for a single treatment. This is entirely not at all possible in case of country like India. So here is the tradition. So what innovation we have done? So for CAR-T, there is a traditional approach, and then next is the next generation approach. So we are adopting the next generation approach. So before going into the next generation approach, I'll discuss few details about the traditional approach. So for traditional, the process starts with the evaluation, followed by the collection, multiplication, conditional therapy, infusion, recovery. And the cost for the treatment goes from the 350 to 750,000 US dollar per patient. Now, if we adopt the mRNA, so all these collection, multiplication, conditional therapy, infusion are no need to be performed, and the cost will be close to a lakh, or maybe a few lakh rupees using the mRNA. Yeah, so here is the CAR-T. So CAR-T is the chimeric antigen T cell receptor. So in case of CAR-T, the T cell receptor, that is the signaling domain, are complex or conjugate recombinant fusion is made with the scFv molecule, which is which is the chimeric antigen receptor molecule. So tumors are categorized into two categories, blood tumor and solid tumor. So the CAR-T therapy which are approved so far are only for the blood tumor, and none of them are approved for the solid tumor. For the blood tumor, identifying the novel target, we use the fast display library, which is a naive collection of the human entire repertoire displayed on the surface of the phages. And that's how, by identifying the best scFv or the chimeric antigen receptor, we reprogram the T cell using these novel CAR. For solid tumor also, we have identified a method that is based on the new antigen. So these new antigens are results of the somatic mutation, and this could be public or private, means it can be same among the individual, or it can be person specific. And for again identifying the best CAR molecule or the chimeric antigen receptor, we use the scFv, we use the fast display library. And for introducing these design CAR into the T cell, there are two approaches. One is the stable and another is the transient. For stable, we use the lentivirus and CRISPR-Cas9 platform. And for transient, we are using the AAV and mRNA platform. Now the third focus area. So mRNA-based treatment therapy for hemophilia A, which is a rare disease. There are a lot of publications, but here I am just highlighting two of them. Correction of bleeding in experimental severe hemophilia A by systemic delivery of factor 8 encoding mRNA, which was recently published. And here also this article was recently published. So people are already trying mRNA platform for curing this hemophilia A. And here is the biology of hemophilia A, so I'll not go into the detail. So we are targeting the factor 8 here. Here is our approach. So we designed the mRNA that is the precursor for factor 8, and with secretion it will get into the mature form, and finally it will go and cure the hemophilia or the disease. Now coming to the summary side. So these are the take-home messages. So Genoa is the pioneer in the field of mRNA vaccine in India. In-house development from bench to bedside we have done for this mRNA vaccine, and one of the vaccine is already approved, and next is at very late stage of development, that is Omicron specific booster. Backward integration to support the Honorable Prime Minister Atmanirbhar Bharat mission we have done. So we make our own own raw materials and own key raw materials or key enzymes. Various mRNA vaccines are in pipeline for infectious disease like malaria, uh, HPV, zoster, and we are working towards the betterment of mRNA platform design and mRNA manufacturing process. The orthogonal approach of establishing the gene editing tools to address the genetic disorders we have taken, and a novel approach towards the CAR T cell therapy to make it affordable also we are doing. And for this, we are collaborating with scientific leaders to address the rare diseases. Thank you.
Thank you so much, Dr. Ajay, for providing us an extremely lucid presentation. I should say you did not waste a single second in providing us information, which is very nice of you. You provided us a comprehensive overview of the mRNA platform. You provided us the capabilities that Genoa already has through their platform processes and the future the company wants to take. And you also touched upon updating us with the key concept also in between. So I really appreciate that. Before I put out the questions, whoever has joined us, if they have any particular question, please raise your hand and we'll take it one by one. Afterwards, I have a couple of questions to ask to Dr. Ajay.
Yeah, please.
Okay, so I think I'll go ahead with my questions. So number one question that I have. So given that you are planning to go for ex vivo sort of intervention using CAR T cells or so I'm my main question is how is Genoa looking at scalability of the nanoparticle or CLNE dose for gene therapy or large interventions apart from just cell-based applications? Because 10 microgram for intradermal at the vaccine level, nanoparticle or CLNE, vis-a-vis cell manipulation in a dish or a small bioreactor vis-a-vis a gene therapy dose, how does the safety profile or how does the scaling of manufacturing of the CLNE nanoparticle is seen by Genoa?
Okay, thank you. So I'll give the justification in two parts. So first is the CLNE. So for CLNE, the key lipid molecule we are making in-house, and recently we have made a new facility for manufacturing of the mRNA vaccine. And this facility has enough space and you can say the capex side to make millions mRNA equivalent to millions of day in a day. And second, I'll say that we have done a couple of innovation right now, and we are getting the data set from animal where there is no need to even have the lipid particle complex with the mRNA. So I mean to say that mRNA can be injected in the naked form inside the body. And if you talk about the dosing side, so we are doing a lot of innovation in the mRNA manufacturing process. And like earlier, we used to make mRNA somewhere, you can say like around 7 to 8 gram per ml. Now we are making mRNA close to 20 gram per ml.
And when it comes to any type of CAR T manipulation using mRNA, what is the kind of typical dose you would need? Is that shareable data?
Those identification part is still under the development. We have not yet finalized the dosing. So once we have the data, certainly we will share.
Thank you, sir. My second question is you mentioned about SAR, your platform, which is self-replicating. Um, I was just having two questions on that. One is the ORF that is present in SAR. That is also you build it in a thermostable manner, or that is a regular ORF that we have for the mRNA with
It's
ribose.
thermostat.
And what is the self-replication time and intracellular persistence for a self-replicating mRNA?
So there are various literature which are published, and they have shown the mRNA expression till day 65, day 69.
So the way I'm looking at it is, I mean, so if there is a therapy, either a CAR T therapy or any type of other therapy platforms, if we have to look at, we must be looking more at repeated dose
Right.
treatment using mRNA platform, and it will not be a one-time dose. I just
Yes,
want to.
yes, because mRNA always go in a transient manner.
Hmm, hmm. Third question I had was mRNA vaccines recently were noted, albeit at a very, very low frequency. They were known to trigger the inflammation of the heart and heart linings. Whether those particular parameters were specifically looked into when you conducted your clinical trials.
Yes, so those kind of practices or those kind of data sets are generally reported from the non-amplifying mRNA platform where people have used sometimes modified bases. But in our study, we have not seen such kind of indications or any adverse event.
Were they specifically looked into in your when you are collecting the safety data? Did you specifically look for any type of biomarkers or parameters or any type of molecular markers that will tell you about heart or heart lining abnormalities during safety profile or lab listings, things like that?
No, not as such. During COVID, we have not done this exercise, but yes, down the line, the molecule that we are having the in the pipeline, we will surely go for those one. Yes, but in challenge study, we have done the histopathology and a lot of biomarker study, but that is again in case of hamster only.
Okay, thank you. Next question that I have is, do you see that this particular platform can be used maybe through an organized system or some sort of any small implantable cell tissue system in a live animal model system, or any, are you doing any type of studies like that?
Sorry, didn't get your question. Can you please repeat?
So yeah, so where the question what I'm asking is, instead of treating the entire organism or an organ, are you looking or are you doing any type of experiments wherein a small subsection of that particular organ, say you can take a small pancreatic tissue or small liver tissue or something like that, take it out, modify it, put it back, and then try to see whether that will do any type of help in eliminating the issues.
So we have not done this exercise so far, but yes, for some of our gene therapy projects are based on the similar uh, way that you are saying. So I mean, the data will come in next couple of months.
Okay. And one more question I wanted to ask is, so do you have any I mean, recently ICMR came up with a proposal, request for proposal, wherein specifically the hemophilia and other things they were not taken into account. But apart from hemophilia or thalassemia, do you are plan to do any other rare diseases in your portfolio as trying to look at how?
So far we have not yet started, but last month we had a meeting at Delhi with the Ashoka University and all the people again from the ORD group. So we are going to collaborate with them, and we will be then working on these disease area as well. Because
Okay.
we are not expert on the disease side, so we need the expert from the academia or from ORD side for this disease model.
So if I understand it correctly, you have also started looking at other different types of rare diseases, probably muscular dystrophies or muscle-based diseases, and then trying to see whether this platform can be used
Yes.
to do this. Yes. Thank you so much. That's all from my side. Those are all the questions. If you have any questions to Dr. Ajay Singh, please do reach out to us. We will pass on those particular questions to you. And thank you, Dr. Ajay. And also, team, there are a couple of your mRNA team has also joined us. online. Thank you so much for joining. And I'm sure, even though right now we have very few people, this particular video will be seen by far more people. And I'm really thankful that this was a very technically interesting uh, webinar that we had. And I thank Genova Biopharmaceuticals as well as Dr. Ajay Singh for coming out and spending a little bit of time with us. Thank you so much, sir.
Thank you. Thank you.
Thank you, everyone. Please do not forget to visit Organization for Rare Disease